Hello, my name is Rika Hirose. I'm fourth year student at Ritsumeikan University. My subject is effect of food viscosity on drug dissolution rate. First of all, I provide you some background on this study. In recent years, the guidelines for bioequivalence testing in the development of generic drugs have changed. The major change was the addition of meals VE test. Therefore, it is important to know the characteristics of the stomach after meals. In this experiment, we focus on the viscosity after meals. The food viscosity affects the dissolution rate of a drug. The viscosity can be classified as micro and macro viscosity. Micro viscosity is observed for the mixture of solvents. On the other hand, macro viscosity is observed for the polymer solutions. Micro viscosities affect both the diffusion coefficients and the unstirred water layer thickness. In contrast, macro viscosities only affect the latter. In this study, we examine whether the FDA meal shows micro or macro viscosity. Next, I talk about the theory of the study. According to the Levitch and the Einstein-Stokes equations, the slope of the log plot of the dissolution rate and the viscosity of the solutions calculated will be minus 1.17 for micro viscosity and minus 0.17 for macro viscosity. Let's move on to the method. The module drug was ethyl para-aminobenzate, and pellets were prepared using a single punch tableting machine. The intrinsic dissolution rate was measured by a static disk method. Test solutions were used sucrose, L-hydroxypropylcellulose, FDA meal, and distilled water. Next, I'd like to talk about the result. Figure 4 shows the effect of viscosity on dissolution rate of ethyl para-aminobenzate. The vertical axis shows the drug concentration, and horizontal axis shows the time. And figure 5 shows the relationship between dissolution rate and the viscosity of the solution. The vertical axis shows the dissolution rate calculated from figure 4, and horizontal axis shows the viscosity of the solution. As you can see from this, the slope of the sucrose was minus 0.68, LHPC was minus 0.2, and FDA meal was minus 0.06. Next, I'll discuss the result. The slope of the sucrose was minus 0.68, which is range of minus 0.17 to minus 1.17. So sucrose was classified as micro and macro viscosity. On the other hand, the LHPC solution's slope was minus 0.2, which is close to minus 0.17, so it was classified as macro viscosity. The FDA meal solution's slope value close to LHPC solution. Therefore, it exhibited macro viscosity and it has little effect on the dissolution rate. The viscosity of FDA meal may be originated from polymeric components such as dietary fibers. Let me conclude this study. The viscosity of FDA meal was classified as macro viscosity. So to evaluate the dissolution rate of drug after food ingestion, a macro viscosity solution should be selected as a test medium. I hope you found my presentation too meaningful. Thank you.